I am delighted to introduce the next speaker, Professor John Fraser from Prince Charles Hospital in Brisbane. Professor Fraser is a graduate of the University of Glasgow, where he received fellowships in medicine and anaesthesia, before he then completed his training in Brisbane and also completed a PhD at UQ. He's a preeminent intensivist at the Prince Charles Hospital, where he founded and leads the multidisciplinary critical care research group. This is the largest group of its kind in Australasia. It has received more than $20 million in grants and industry funding, and the group was awarded the Centre for Research Excellence in Australasia, looking at the development of bionic hearts and lungs.
Thank you very much for that kind introduction. And I'm probably one of the opposition. I'm not the scientist coming to talk about how we can do with medicine. I'm the medic trying to talk about how we can integrate with the proper scientists in the room. I'd like to thank this society for giving me the opportunity to present the work that our group is doing. So I'm a clinician. I do 70% of my time as clinical service, and I look after intensive care units and therefore the sickest patients in the hospital. And what we're seeing is a tsunami of heart failure across the world, not just Australia, but everywhere across the developed and soon to be the developing world. $10 million US per year easily, 25 million lives by 2020, and probably greater as the diabetes epidemic gets worse and worse. And at least in the hospitals in Australia, the wheels are starting to fall off the system because we really can't cope with the magnitude of disease burden or the severity of that burden. So if we look at why we think it's going to get worse, we've seen a lot of eminent journals. I went to the eminent journal of the Weekend Australian and looking at the changing demographics of Australia from 1925 to 2000 and to 2045. So we call this from the pyramid to the coffin. And in the 1925 time, most people died before they were the age of 50. By 2000, society, a little bit like myself, had started to spread out a bit round the middle. And by 2045, more and more people are going to be over the age of 60. And as medicine gets better and all these fantastic devices we're talking about gets better, patients are going to be later and later on, and that burden of cardiac and cardiorespiratory disease is going to get bigger. So what do we do with patients with heart failure? I'm not sure how many will have seen patients with heart failure, but it's a pretty horrible death. You can't breathe properly, you can't walk to the toilet, you can't sleep, you're exhausted, but you can't really sleep because you just can't get a breath. So it's really not an option to do nothing to these patients and let them turn up their toes. We can use the whip, so we use drugs to increase the contractility of the heart. The problem with these drugs is it increases or speeds up the death. So the muscle's damaged and we make it work harder. It's counterintuitive. Patients do actually say to you, if you give me 6 months of good life versus 2 years of bad life, I'll pick 6 months. But this is not actually curing the problem, it's treating some of the symptoms. We can reduce the load, so the heart is pumping against an afterload, pumping against the pressure, and we can drop that pressure that it's pumping against by changing the neurohumeral cascade by using devices like balloon pumps and different drugs. But again, it's a copious quantity of drugs. They do work. They have got a vast cost associated with them, and they can cause other organ dysfunction. Or we can fix the shoes. So if the heart failure and the disease is happening because the blood vessels are blocked, we can open them. If the valve's dysfunctional, we can replace the valve, either open surgery or, as we're doing much more now, percutaneous surgery. Or we can resynchronize the heart using the pacemaker type devices that you've seen, where we can resynchronize both chambers. We can heal that donkey. That donkey might be sick, and we'll talk a little bit about some work that Nathan Palpant from UQ is doing with our group in cardiac stem cells. Or if all else fails, you can really take the sick donkey and get a new horse, give it a transplant. And this is Farlap, and this is the heart of Farlap. They tell me it is a massive heart, and I think it's in New Zealand at the moment. But if we look at changing the donkey into this horse, it sounds fantastic. But the problem is the number of donors. If we take a diagrammatic representation of the number of patients who have got end-stage heart failure that could benefit from a transplant, it's massive. But the number of patients we actually assess for a transplant is tiny. Why? Because we know there's no point in assessing people that we will never be able to give them a transplant, because there's specific criteria for the transplant organ that we just can't meet because there's not enough donors. And even some of the donors that do want to donate, the heart and or the lung tissue they want to donate is too damaged in the act of brainstem death that it is not transplantable. And from that group that we assess, a tiny fraction are transplanted. So I think 70 transplants in Australia last year, that's all. It's interesting looking at how different countries work in different areas, and I presented a talk in a similar area in the States, and the day before I presented, it was talking about putting one of these ventricular assists into Dick Cheney. And I was talking about brainstem death and how different drugs can affect brainstem death, and I said, but America, you people do things different. And there was a couple of nods. I said, you really push the envelope here. They nodded. You know, you just do it differently. They were up in the halls, they were waving their hands. I said, because in Australia, it's our donors that have to be brain dead. But here, it's your recipients that are brain dead. That was the last time I was invited to that. So. But we've got Mr. Trump coming, so moving on. It's Canberra, it's apolitical. So, getting a new horse would be ideal, but we probably can't do it. So what else can we do? We can get something else to lift the donkey until it's fixed. And that's really the idea of putting it in the back of a tractor. Apologies for this picture, but this chap actually just phoned me 10 minutes ago. This is a farmer friend of ours who was working a $6 million property and developed heart failure. We couldn't transplant him. He employed hundreds of people, and we just could not get a transplant. The problem of not getting a transplant when it's a heart is that when the heart stops working properly, it stops supplying oxygenated blood to the kidneys, the lung, the liver, the brain. And bit by bit, all these organs that are probably quite functional start to peter out because they're not getting enough oxygen. So once you get to a certain level of other organ dysfunction, you are no longer transplantable. So if we can't support the heart, you fall off the transplant list. No matter how good your organs were beforehand, you fall off because we've not been able to give you something in time when you need it. And these are some of the devices we'll talk about later on. But these are the sort of massive operations. This was about 12 years ago. He's still doing very well, that we did. And this is the amount of blood transfusion this man required in the 3 weeks prior to transplanting him. Vast amount of drugs, vast cost in the intensive care, huge risk of infection, and a huge cost to society and the blood donors and the other operations that couldn't proceed because the 165 bags of blood were used in one man. This is him now, and he actually raised $75,000 for us. He owns half of Queensland, and he did 24 hours wheat planting and won a Guinness World Record for it, John Corrigan. So we've got a big problem with sick hearts, but also sick lungs. And we heard a lot about Scotland, and you may detect a slight hint of an accent. I would say that I don't have an accent. I would say it's the Australians that have the accent. But equally, I just thought it would be worth pointing out how long ago we started supporting organs with mechanical devices. And this paper was actually 40 years before Edison got his light bulb working, that a Scottish chap called John Dalziel developed the first ventilator, negative pressure ventilator, which was worked by a bellows. So even back in 1838, before electricity, people were looking at how could we mechanically support organs. It was tricky because there was no electricity, so clearly the medical student on the side was pumping those bellows for quite some time. Things advanced with ventilators, and these are the Curas ventilators in the States and California, where it was a negative pressure ventilator, so basically the chest wall would be sucked out and closed down, sucked out and closed down. It was fascinating. It kept people alive through the polio epidemic, but at huge physiological cost and wasn't particularly effective. Was interesting. I thought this was a fairly telling cartoon. This poor man's got polio. He's completely paralysed, and just after he's been paralysed, this nurse has the bad humour to tell him that there's a vaccine just available for polio, but he's just too late for it. So she's still got a not particularly happy smile on her face either. So we've moved from those Curas ventilators on to much, much more critically ill patients, but we are dealing with patients now that would have long died even before they left hospital. And this is from our animal lab. These are lungs that don't work as a single compartment. So the area of lung here is quite nice and pink. This is what lungs meant to look like. But this area here, and this is an acute lung injury using smoke inhalation, is completely damaged and atelectatic. So if you imagine the driving pressure you need to open an area of lung here and the area of lung here, they're completely different. They're completely different compartments. But all we have is a breathing tube down here, and we blow air in at a certain pressure with a certain oxygen content, and the pressure required to open this lung is way too much for this area, and it damages that lung. And sometimes what we actually have to do is we use 2 ventilators and 2 tubes, so we ventilate lungs independently of each other. But again, that only works if it's just a lung to lung rather than anterior part of the lung to the posterior part of the lung. And what we do when we do these these types of ventilations with very sick lungs is we actually force the lungs to work hard. We use higher pressures, higher opening pressures to try and keep them, but we're actually making damaged organs work harder. Scotland revolves around football, and the analogy that I'd use here is this is a man who, for the non-medics amongst you, has broken his leg. But who would ask this man or expect this man to keep going playing football with this broken leg? What you'd expect to do is to take him off the pitch, put him in plaster of Paris, maybe do some bone grafting or some polymers into him, and let him rest for 6 or 8 weeks until that leg's broken. But what we do with the sick lungs until we started using the more modern mechanical devices is really just working them harder and harder, and that's why we had such a significant mortality. So we need to use different ways of moving away from the ventilators and things. So the subjects we're going to talk about are ECMO and ventricular assist devices. ECMO, extracorporeal membrane oxygenation, it's a short-term device, normally plumbed into your leg with large pipes. It can be veno-venous, which means the blood is still pulsatile and it just supports your lungs, or veno-arterial, where it's non-pulsatile and it supports the heart and or the lungs. It can be put in an emergency. If you're slick at it, you can put this in a cardiac arrest situation. A number of hospitals around Australia have got good survivals when the heart is actually stopped. Someone's doing CPR and someone's cannulating the groin, ensuring adequate perfusion to the brain and the other organs. With the ventricular assist, these are long-term pumps that are implanted, can be used left or right heart. Not really very good in the emergency situation, very, very expensive, 100,000 plus, and generally non-pulsatile, and we'll explain that shortly. So to go to ECMO, it really came out of cardiopulmonary bypass. It's been around for a long time with Zappel in '68. And essentially, blood comes out of the system, blood comes out of the body, goes to a heater cooler, oxygen's put into the circuit via an oxygenator, and there's a lot of technology around this, and carbon dioxide removed, and at a normal thermic temperature, the blood is reinstituted back into either the right side of the heart in respiratory ECMO, or the left side of the heart when you're using cardiac support. And that's just a graphic there. The oxygenators have come on tremendously. They were incredibly non-biocompatible and becoming much, much biocompatible, but there's still a huge amount of inflammation excess that happens around this plastic polymer membrane, which allows oxygenation. Things have improved dramatically. This is a picture from the 1970s, or early 1980s, of a patient on ECMO. And you might notice there's something missing. There's just so much equipment that the patient can't even fit in the Photoshop. There's just equipment after equipment after equipment. And not surprisingly, at these stages, the mortality was excessively high. It's interesting, I think we've seen a number of Hollywood films and a number of ideas. Wouldn't it be fantastic if we all had the budget of Hollywood and we wouldn't have to show Hollywood up there and we could show our science? But same in the cardiac idea. We need an alternative to artificial heart, to lungs and hearts. We need artificial hearts. And as I say, Hollywood, like one of the preceding speakers, has a large number of films about the artificial heart technology, and we need to catch up with them. These are being used clinically already, and if we look at the increasing use, these are ventricular assists, and this is up till about 2012, I think. Increasing use is going exponentially, and this has happened. What used to happen was the ventricular assists would just be used short-term till a transplant became available. Now, with the improved engineering and the biocompatibility side of things, ventricular assists can be used long-term. So if you're not going to get a transplant, you can be given a ventricular assist and say, this is the end of what you're going to get. And these devices, the more modern ones we'll talk about, will last 5, 10, and 15 years. So their use is going up rapidly. And again, the same with ECMO for artificial lung support is also going up rapidly, and the survival is improving, but it's still not good enough. And I think it's because of the imperfect technology and the integration of the bio and the electronics, as we're talking about the bionics. So what we've tried to do, and again, echoing a number of the previous speakers, we're at Prince Charles, which is the largest cardiac hospital in Australia. We had nothing like this before I started the group. And what we've done is we've brought the engineers out of the dungeons of the engineering hospitals and parked them about 200 yards from the operating theatre, about 500 yards from the animal lab, from QUT. And our patients come and speak to the engineers, and our engineers go and speak to the patients. So when we have simple things like lines for the battery pack, our patients haven't really been involved in the planning of it. One young girl I remember came and spoke to us, said, I can't go to the toilet, the cable's too short, which maybe we wouldn't think of. Maybe we were thinking of different aspects of why that cable's that length to minimise the torque in the skin, etc. But we have to engage the patient as a key contributor in our research. And what we're trying to do is get the engineers and the doctors to speak similar languages so that we can come out with devices that are useful. Before we bridge the gap of going into a company, we need to bridge the communication gap between the engineers and the medics so that the medics can explain what we think we need and the engineers can explain the way we're doing it is wrong. That's what they're very frequent and very happy to tell us that. So in terms of mechanical support as it exists at the moment, we have left ventricular assist and total artificial hearts. We tried initially with biological mimicry. The idea was if a heart beats and a bird's wings flap, then we should copy the beating heart. But we know from the early versions of the aeroplane that aeroplanes with wings that flap are not very good aeroplanes to go on. And the pumps that we started with, first generation ventricular assists, were pulsatile devices. They were essentially a balloon, a 65 ml balloon in a sac, a bladder, which would fill with blood, attached to a pneumatic driver. Once it was filled, the new driver would send a signal back to the chamber and it would eject the blood. It kept people alive, but it would perish. It wouldn't last for more than about a year. It sat outside the body paracorporeally with two pipes per pump, where there's a huge risk of infection and also lots of red cell damage. But bit by bit, people have moved on to realise we don't perhaps need the pulse, and we can exist without a pulse, and therefore biological mimicry is much less necessary. These are the first, second, and third generation pumps that we were talking about. The first generation was the Thoratec. Second generation still is a bearing, and third generation, the types we use across Australia now, types like the Heartware and the Heartmate 2, have no bearing, so that means there's much less blood cell damage. So I've gone through this a little bit. Lots of clots, lots of infections and short-term support. The second generation, these were the ones that started to have no pulse, but have the problems of having a bearing there, and you would start to get some red cell damage. With the third generation devices, essentially there's a magnetized impeller. It's run by magnetic levitation, and it's suspended on blood continuously, so there's no valves to block off, and there's no moving parts. It's really just coupled with the electromagnet on the outside and suspended like a surfboard and some blood. The total artificial hearts are way behind at the moment. This is the AbioCor, which was, excuse me, licensed but way too big to get in any patient other than a very, very large man. Syncardia, again, pulsatile, same idea as the Thoratec, the pulsatile device. Essentially two pumps stuck together inside a chest with a pneumatic driver that was about the size of a washing machine. Carmat is also a big one. This man's got 7.5 gloves, so that's going to be a big person that's going to fit into. And then Cleveland Clinic and Bivacor, a group I was involved in, have got the total artificial hearts being looked at. So the question is, at the moment, we don't have a good total artificial heart. Should we use two of these LVADs, which are licensed just for the left side of the heart, or should we try with one of these big total artificial hearts? So the analogy I guess I'd give here is that if we use two of these left-sided pumps, the spinning disc pumps, we can put one in the left side, which is developed for left-sided pressure, and then change the electronics to its supports on the right side. But if you do put two in, what you need to do, you've got two independent pumps that you need to keep running exactly the same speed. It's a little bit like tying two motorbikes together and keeping wheel by wheel aligned all the way to Sydney. So there's major problems can happen with it. But we've done this in the animal lab and it works well, and people have started to do it clinically until we get a good total artificial heart. The idea with total artificial heart is either replace it with one device or assist it with one device that talks to each other. And this is the Bivacor, which is the brainchild of a chap called Daniel Timms, who did his PhD at QUT, and then we developed a company out at Prince Charles, and he's now taken that over to the States. It's a lot smaller than any of the other ones you've seen. It's got electromagnets here and axial magnets here. It can replace or assist the heart. The heart can be removed, or it can be plumbed into it. Now's the moment. This is the device, one of the previous iterations of the device. So the axial magnets at the bottom, the electromagnets at the bottom, coupled with the impeller inside. So the blood comes back deoxygenated from the right side of the body and is pumped back out to the lungs, picks up some oxygen, goes to the left side, and then pumped up to supply these other organs. And the impeller in the center has got shorter blades for the lower pressures on the right-hand side, longer blades on the left-hand side, and it can adapt, when it's being used as an assist device, it can adapt to support the right heart more or the left heart more, depending on which side is more dysfunctional. Or it can be used as a total artificial replacement, as we've seen before, because heart failure is a fairly dynamic creature. And this is what the size, the relative size, this is just a dummy version, this is the relative size versus 2 of the other ones, and this will do the exact same job, but implanted internally. We did some work last year, and we did an implant. This took a great amount of work from Daniel, the team, and various people from across the world did this. And this is one of our animals with no heart, a little bit of atrium that we sutured the device into, and a very happy and healthy sheep, and ate lots and lots and lots. And this is Daniel on the front cover of the Australian magazine, and the whole team was there for about 4 days continuously. So mechanical support is here already, and it's coming and it's getting better, but there's a whole pile of bits that we don't understand of the integration of the body and the device. The coagulation and inflammation, what happens to drugs when you put a semi-permeable membrane like an ECMO circuit and you give antibiotics to kill the bacteria or to kill the swine flu. So the device keeps you alive, but paradoxically it drops your ability to kill the bacteria or the virus. Lots of blood being transfused, the inflammation, etc. And how do we image metal things in our body? We know how we can image hearts with echoes or chest x-rays. How do we do that with these devices? Massive amount of problems with them. And if you look, this is a study of 3,000 patients. And essentially, if you look at them, the big events are bleeding, infection, neurology, and almost everyone's getting one. So we are doing well, we are keeping patients alive, but we've got to do better. We've gone to the animal lab from the ECMO side of things as well as the artificial heart side of things. This is an NHMRC project that we had, looking at all the different aspects from how too much oxygen in the blood can cause problems, then ex vivo, then a healthy animal, then a sick animal, and then onto humans, and incrementally approach it to understand the mechanisms and optimize the therapy. So we need to integrate the physiology and pumps, and it's great that so many people have spoken before really about the integration of the body and the devices. Other areas that we're working at is these lungs that we can't transplant. So we've talked about the artificial things, but we also want to optimize the lungs and the hearts that we can transplant. So this 50% of lungs cannot be used for transplant because they're too damaged in the active brainstem death. So working with the group 16 in Norway, this is a rig where we can take damaged lungs out of someone that wants to donate, plumb them onto this rig, resuscitate these lungs on this rig, optimize their function, optimize their oxygenation, and use lungs that would not be transplantable 12 hours later are transplantable. We've done this in the animal, and we've also done it in humans, and it works very successfully. And on top of this, we are other groups are looking at stem cell integration within these lungs. Again, the same idea with the heart. We have many hearts that want to be used, but Australia is a massive country. So if you imagine a heart's you don't need to imagine, the heart is a big muscle. It takes a huge amount of oxygen out. So essentially when we take a donor heart, we go to Mount Isa, we see the donor, we make sure everything's done, we open the chest, take the heart out, put it in a bag of ice, and essentially run really, really fast. Because from the time, and it put in a bag of ice, which is not particularly brilliant technique, but it's what we've got at the moment. So every second that heart is outside the body, the cells are starting to decay. And after about 4 hours, it becomes what's called a marginal donor. And the risk of that heart failing when we put it into the recipient becomes more and more, and the function becomes less and less. So remember, when we take, this is a no going back scenario. We've got a patient, we've got your mum, we've got your dad, they're alive. Once we take that heart out and put this heart in, we can't go back and switch it back out. So what we're trying to do with this ex vivo rig is resuscitate the donor as well as possible. Have a rig on standby at the donor bedside where we can actually, I think it's here, where we can actually perfuse, this is just cordial or something, we can actually perfuse the heart with oxygenated blood with a variety of different mediators that we're trialing, plus and minus stem cells, so that by the time you've flown from Mount Isa or Darwin to Canberra, the heart's almost as fresh as when it left the patient, and that your mum and your dad is about to get a heart that's basically as fresh as a beating heart. And we've got some work started in this too. Being Scottish, I think the Northern Irish colleague will get the importance of the Celtic project. It's actually called the Celtic project. But the other problem with extracorporeal membrane oxygenation is there's a huge amount of inflammation induced as our activated, inflamed red cells, neutrophils, platelets hit this plastic membrane. So there's some studies looking at ECMO with stem cells, mesenchymal stem cells in acute lung dysfunction. We now think we can incorporate them into the oxygenator. So we've just started this PhD as part of our Centre for Research Excellence and also with Palpant's lab at UQ to try and see the inflammation and the deterioration of the patient as they're put on ECMO. Can we ameliorate this with use of MSCs? And the Queensland Government's been very kindly giving us some money for this nice glossy brochure. And we're starting to work with cardiac stem cells as well, and this is Nathan Palpant's work. So not just stem cells, but stem cells, as you'll see, that are starting to work as a syncytium, starting to pulse together. Only 3 weeks ago, the first big, good study, I'd say, in the Lancet showed post-infarct with heart failure, injection of stem cells into the failing heart can actually improve the pump function and reduce mortality. So our plan now is we'll start doing some models. We've got models of heart failure in the animal. We'll support the sheep, we'll support the heart with the tractor, with the new fancy whiz-bang mechanical device, but equally integrate stem cells whilst the ventricular assist is being the tractor, and we can start to try and regrow some myocardial tissue within it. And again, Nathan's got some nice early work on mice as well. So it's really just trying to integrate the biologics with the stem cells and the mechanical devices. So is it prime time? This was 1984, one of the first pumps that was used. I think everyone thought at the time we were just around the corner. We're definitely making people survive that wouldn't have survived in the past, but the mortality is too high, the financial cost is too high, the physiological cost is too high, and I think it's because imperfect technology, poor integration, and that's what our group and other groups are trying to achieve. And as everyone else has said, the synergies from interdisciplinary work are the key to it all. Thank you.
Thank you very much for a fascinating talk and very graphic slides. It reminds me why I could never be a doctor. Questions?
Thank you, John, for that fascinating talk. My question is, it's a very naive one, but the human body, I guess, is used to a pulsating sort of temporal variation of blood pressure. And so with the rotary heart, which produces a steady blood pressure, I guess, will there be any problems?
So I don't know, but we're investigating it. We don't know. It seems to be, so once you get down to the capillary where oxygen is actually transferred, there is no pulse. So when the actual machinery of the body is not getting a pulse. However, I think in the big blood vessels, there's an endocrine effect of the pulse. So the endothelium releases a whole pile of proteins, and I think it probably does respond to pulse. So we've got a number of projects looking at also what happens to the shear stress with a pulse and without a pulse, looking at platelet activation, because clotting in these things is a major problem. Platelets are not just clotting, they're actually a huge initiator of inflammation. So if you come to our lab one day, we'll have the other thing we can do is, because we've got so many clever engineers, we can change these pumps programs so we can actually pulse them as well. So what we can do is take a non-pulsatile pump, change its programming, make it pulsatile by just changing the speed of it, and in one bath have fresh whole human blood with a pulse, and in the other blood, so you'll see lots of our students walking around slightly pale with a sticky plaster on here because it's fresh. And we can look at the changes in the blood. We're also looking at the changes in the skin. Skin's the largest organ, and most of these patients will get pressure areas. So the skin's right at the end of the line in terms of the pump, and if anyone from the country, you talk about a brownout, not a blackout, there's just this incredibly low flow. What happens without the pulse there? We don't know. We know that in the clinical studies, patients with the newer pumps have a higher survival, but do tend to get more bleeding from the gut, and that's probably, again, the shear stress and the cutting of the platelets. But a lot of those questions are not known, and that's what some of the studies were also doing.
I'm interested in the dynamic control. So if the person needs more oxygen and they need more blood flow, how does this heart know about that, and what's the technology around there?
So, good question. The old techniques, the pulsatile devices, were actually slightly more physiological because they had a Frank Starling mechanism. So the pump would only pump once it was full. The newer phase 2, second generation pumps, etc., would just go at the set speed. The doctor would set the speed, and there you go. And if you talk to our patients, if you'd set it at 5 or 6 litres, that would be enough to go maybe walking down to the other house we went to when the fire alarm went off, but way too much for lying in bed. So they tell you about lying in bed with all this blood pumping in. So there's a whole pile of active and passive physiological control mechanisms we're working at. We can use the amount of blood returning back as a feedback mechanism to try and use sensorless. And then there's also with a passive control system built into the cannula that when we're using the pumps which go into our heart, they actually have a contractile part to stop suck down, and it will also change the speed. Again, we're also trying to do some work on coronary and cerebral autoregulation. When these pumps are in the heart, we're hoping they're going to recover, and we're talking these pumps going to 30-year-old women and men. We want the remaining heart tissue to be as well oxygenated as possible. So we try and make sure that when we do put it into these patients, we can optimize the blood flow down the coronaries, up the carotids. But it's a big area of work, and there are active and passive control mechanisms that we're working on simultaneously.
Any further questions? If not, please join me in thanking Professor Fraser.